Okay, so let's dive in. If you think about the whole body, all the systems working together, like this huge, complex orchestra,
Mhm.
then the anterior pituitary gland, well, it's basically the conductor, wouldn't you say? This tiny little thing, the adenohypophysis.
Absolutely. It's setting the tempo for almost every other endocrine gland. We're talking
Oh.
growth, metabolism, reproduction, how we handle stress. It's central.
Totally. And today, we're really going behind the curtain. We've got some detailed medical and histology sources here, and the plan is to get into the nitty-gritty, the microscopic details of this vital organ.
Yeah, exactly. We want to understand the actual cells doing the work, the specialized players, and also how it's all controlled.
Yeah.
The regulation is incredibly precise. We need to see how the conductor keeps everyone in time.
Okay. Let's start with the physical structure, the anatomy, the whole gland hanging off the brain. That's the hypophysis.
Right.
But it's the front part, the adenohypophysis, the glandular bit, that's really doing the hormonal heavy lifting.
That's the one. And that glandular tissue is not just one blob, it's divided into three sort of distinct areas. The big one, the main player, is the pars distalis. Makes up about, what, 75%?
75%. Wow. So that's the main factory floor then.
Pretty much, yeah. That's where most of the hormone synthesis and secretion happens.
Okay, so what about the smaller parts?
Well, you got the pars tuberalis. It's like a thin sleeve of tissue that wraps around the stalk. The infundibulum, connecting the pituitary to the hypothalamus upstairs.
Ah, okay, the connection point.
Mm-hmm. And then there's the pars intermedia. It's this narrow little zone, kind of sandwiched between the anterior lobe and the posterior lobe, the neural part. Often overlooked functionally, but it does have some interesting quirks we should probably touch on later.
All right, pars distalis is the main stage. Let's zoom in on the performers there. Our sources say there are 5 main types of hormone-producing cells.
5 major endocrine cell types, yeah. And what's key is that they're all making and releasing protein hormones.
Okay, so who are these 5? Can we group them?
Yeah, we can think about them functionally. Maybe start with the ones handling, like growth and basic bodily maintenance.
The maintenance crew. I like it.
Right. So first up, you have the somatotrophs. They make growth hormone, GH, and actually they're the most numerous cell type in there.
Most common. Okay.
Then you have the lactotrophs, sometimes called mammotrophs. They produce prolactin, PRL. That's the hormone involved in milk production.
Got it. Growth and milk. Simple enough. What's next? The ones managing other glands.
Exactly. The coordinators.
Yeah.
So you have the thyrotrophs. They produce thyroid-stimulating hormone, TSH. That's the signal telling your thyroid gland to, speed up or slow down your metabolism.
Makes sense. And the other coordinators, the gonadotrophs. These are pretty interesting because they actually produce 2 different hormones, both glycoproteins, follicle-stimulating hormone, FSH, and luteinizing hormone, LH. 2 from one type of cell, or different cells.
Well, the sources suggest it might be the same cell making both, maybe depending on the specific signal it gets from the hypothalamus, or perhaps distinct populations. It's still debated a bit.
Fascinating. Okay, that's 4. What's the 5th? And you mentioned maybe the most complex one.
Yes, the corticotrophs. These guys are biochemically quite sophisticated. They don't just make one hormone directly. They synthesize this really large precursor protein.
A blueprint, you called it earlier.
Yeah, exactly. It's called pro-opiomelanocortin, or POMC for short. Think of it like a multi-tool protein. It doesn't get released as is. Instead, the cell cleaves it. It cuts it up into several smaller active peptide hormones.
So one initial protein leads to multiple signals downstream. Why?
Efficiency, probably for stress response. The main product cleaved from POMC is adrenocorticotropic hormone, ACTH. That's the big signal to your adrenal cortex to release cortisol and manage stress.
Okay.
The cleavage process also produces things like beta-leaky lipotropin, which is itself a precursor for endorphins, the body's natural painkillers.
Wow. So when you're stressed, the same cell type releases the stress signal and something to help manage the pain or fallout, all from one starting molecule.
Precisely. It's a pre-packaged survival kit, essentially. Very efficient design.
Incredible. And just quickly, are there any other cells in there besides these 5 hormone producers?
Yeah, good point. There are the non-hormonal cells, the folliculostellate cells. They're kind of the support crew.
Like glial cells in the brain?
Sort of, yeah. They provide structure, help manage the local environment around the endocrine cells, maybe even help them communicate with each other through paracrine signals. They don't make the main hormones, but they make the whole operation possible.
Okay, so we have the workers and their products identified. But if you're actually looking at this tissue, say under a microscope for pathology, how do you tell these 5 types apart? They must look pretty similar, right?
That's the challenge. And historically, the first way to tackle this was using basic staining, like hematoxylin and eosin, H&E staining. This doesn't tell you what hormone is inside, but tells you about the cell's general state, especially its granules.
Okay.
So H&E divides the hormone-producing cells, the parenchyma, into two broad groups based on how they take up the stain.
Ah, the chromophils and chromophobes. I remember those terms.
Exactly. The chromophils are the color-loving cells. They stain intensely because they're packed with secretory granules full of hormones. These are generally the active cells. They're large.
And the chromophore.
They're the color-fearing or faint-staining ones. They're smaller, have fewer granules. They might be cells that have just released their hormones, or maybe they're resting, or perhaps they're even undifferentiated stem cells. They're much less common than the chromophils.
Okay, so active versus less active, roughly. But within the active chromophils, there's another classic division, right? Based on the type of color.
Mm-hmm. Acetophophils and basophils.
Mm.
This starts to give us a hint about the type of hormone being made.
How so? Well, the acidophils, like the acidic eosin stain, appearing pink or reddish, these are typically smaller cells, and they're the ones secreting the two simpler polypeptide hormones, growth hormone, GH, and prolactin, PRL. Okay, GH and PRL are acidophils. And the somatotrophs making GH are super common, you said?
Yeah, somatotrophs are the most abundant acidophils, making up almost half the cells in the pars distalis overall.
Wow. Okay, then the basophils must be the other group.
Right. They prefer the basic hematoxylin stain, so they look bluish or purplish. They tend to be a bit larger, more granular. And these are the ones responsible for the four more complex glycoprotein or polypeptide hormones: TSH, FSH, LH, and ACTH. Interesting. So structure relates to stain preference, relates to hormone type. And you mentioned thyrotrophs. The TSH guys are actually quite rare.
Yeah, they're the least abundant of the chromophils. So influence isn't always about numbers in the pituitary.
Okay, so we see these cells, acidophils and basophils. How are they arranged? They're typically found in clusters or cords, sort of like little islands of cells. And critically, these cords are right up against sinusoids.
Sinusoids. Those are
Right.
special capillaries, right? Leaky ones.
Exactly. Fenestrated capillaries, part of that portal blood system, super permeable. This means when the cells release their hormones through exocytosis, the hormones go directly into the bloodstream to be carried away. There's no delay.
Makes sense. Direct deposit for hormones. But just knowing a cell is, say, a basophil, that still doesn't tell you which of the four basophil hormones it's making. Does it TSH or ACTH or...
No, absolutely not. Staining color gets you partway there. It narrows it down, but it's not definitive for function. That's where modern techniques become essential.
Like the pathologist's secret weapon. You got it. Immunocytochemistry, or IHC. This is way more specific. We use antibodies that are designed to bind only to a specific hormone, like GH or ACTH. So you can tag the actual product inside the cell.
Precisely. The antibody usually has a label we can see under the microscope, like a fluorescent tag or something that causes a color reaction.
Sure.
So IHC lets us say with certainty, okay, this cell is a somatotroph, because it's lighting up for GH. Regardless of whether it looked pink or faint with H&E, it's crucial for diagnosis, especially for pituitary tumors.
And does IHC reveal anything about where these specific cell types hang out within the pars distalis? Is there a pattern?
Yes, it does reveal some interesting regional distribution. For instance, the somatotrophs, the GH producers, tend to be smallish, kind of oval-shaped, and they often cluster in the lateral wings of the anterior lobe.
Okay. Out to the sides. What about others?
The thyrotrophs, the TSH cells, they're often found grouped together more towards the front, in an anterior wedge area.
And the gonadotrophs, FSHLH.
Those seem to be more evenly spread throughout the pars distalis, but interestingly, they are the main cell type you find up in the pars tubralis, the part wrapping around the stalk.
Huh. Okay. So speaking of different regions, we need to go back to that middle zone, the pars intermedia. You said I had corticotrophs, like the pars distalis. It does contain corticus, cells that express that POMC gene. But here's the twist. In the pars intermedia, they seem to cleave that POMC protein differently, a different cutting pattern.
Yeah. Instead of prioritizing ACTH for the stress response like the pars distalis corticotrophs do, the pars intermedia pathway generates smaller peptides. Primarily, two forms of melanocyte-stimulating hormone, MSH, and also things like beta-endorphin.
MSH, that affects skin pigmentation, right?
Yes,
That's the known function, yeah. It increases melanin production. So the body uses the same starting block, POMC, but processes it in two different locations to get different functional outputs. ACTH for stress in one spot, MSH possibly for pigmentation or other roles from the middle zone.
Amazing flexibility. But what's the point of MSH in adult humans? Is the pars intermedia really active? That's the million-dollar question. Our sources indicate it's quite prominent during fetal development, but in adult humans, its structure is less distinct, often with cystic changes, and its overall function isn't entirely clear. It might be mostly vestigial, or perhaps it has subtle roles we don't fully appreciate yet. It's one of those intriguing gray areas.
Okay, so we have these specialized cells. We know what they make, roughly where they are, how to identify them. But they don't just decide to work on their own, right? There has to be a higher command.
Absolutely. The CEO of this whole operation is the hypothalamus, sitting just above the pituitary.
Right. And it communicates how?
Through hormones, specifically small peptide hormones produced by specialized neurons in the hypothalamus. These neurons release their hormones into a unique capillary network in the median eminence.
And those hormones then travel directly down to the anterior pituitary through that special blood supply we mentioned earlier.
Exactly, the hypothalamic hypophyseal portal system. It's like a private express highway.
Why is that direct route so important?
Well, it ensures the hypothalamic signals reach the pituitary cells quickly at high concentration without getting diluted in the general bloodstream or broken down elsewhere. It allows for very precise, rapid control. The pituitary gets the message loud and clear, instantly.
Got it. So what kind of messages is the hypothalamus sending down this highway? Broadly, two types. Releasing hormones, which basically say go, secrete your hormone, and inhibiting hormones, which say stop, hold back.
Oh.
Okay, let's cover the go signals first. The releasing hormones.
Yeah. We have TRH, thyrotropin-releasing hormone. That tells the thyrotrophs to release TSH.
Makes sense.
Then GnRH, gonadotropin-releasing hormone. This one stimulates the gonadotrophs to release both FSH and LH.
One signal for two hormones again? Okay.
Then there's GHRH, growth hormone-releasing hormone. Pretty straightforward. Tells the somatotrophs to release GH.
Okay.
And finally, CRH, corticotropin-releasing hormone. This is a big one. It signals the corticotrophs not only to release ACTH and battle LPH, but also to synthesize more of the POMC precursor in the first place.
Wow, CRH really kicks off that whole stress cascade we talked about. Okay, so those are the accelerators. But you need brakes too, right? Especially for powerful hormones like growth hormone.
Absolutely crucial.
Mm.
Constant stimulation would be disastrous. So the hypothalamus also sends inhibitory signals. And the nature of these inhibitors is quite interesting.
How so?
Well, first, there's somatostatin. It's a peptide, 14 amino acids long, and it acts as a powerful brake on the release of both growth hormone, GH, and thyroid-stimulating hormone, TSH.
Okay, one inhibitor hitting two different cell types.
And the other major inhibitor is chemically different. It's dopamine.
Dopamine, like the neurotransmitter involved in reward and movement.
The very same molecule. But here, acting as a hormone delivered via the portal system, its primary job is to strongly inhibit the release of prolactin,
Yeah.
PRL, from the lactotrophs.
Wait, so prolactin release is mainly controlled by stopping an inhibitor, not by a releasing hormone.
Largely, yes. Dopamine provides constant tonic inhibition. Think of it like the parking brake is always on for prolactin secretion. You need a specific signal, often related to pregnancy or suckling, to release that brake, reduce dopamine, and allow prolactin levels to rise.
That makes a lot of sense physiologically. You wouldn't want prolactin secretion running rampant all the time, only when needed for lactation. It's about energy conservation and reproductive timing.
Exactly. It's all about maintaining balance, homeostasis. This tiny gland, the anterior pituitary, is orchestrating so much metabolism, growth, reproduction, stress response by having these very specific cell types tuned to respond precisely to these chemical signals, the releasing and inhibiting hormones delivered directly from the hypothalamus.
It really is the master gland, isn't it? Dictating terms to the thyroid, the adrenals, the gonads. And maybe here's something interesting to chew on as we wrap up. Think about those inhibitors again, somatostatin, peptide, and dopamine, a modified amino acid. Okay.
The body uses these quite potent, often complex releasing hormones, TRH, GnRH, CRH, GHRH, to turn things on. But for two incredibly potent and metabolically significant hormones, growth hormone and prolactin, the primary control mechanism relies heavily on inhibition.
Yeah, somatostatin holding back GH and TSH and dopamine constantly suppressing prolactin.
Right. So why might that be? It really underscores that turning things off or keeping them suppressed until absolutely needed might be just as critical, maybe even more critical for overall stability and long-term health as the signals that initiate hormone release. The brakes are just as important as the accelerator, perhaps even more so for these fundamental processes like growth and lactation. It's a testament to the sophisticated checks and balances built into our endocrine system.
Fascinating point, the power of stop signals and keeping everything running smoothly. Lots to think about there.